The deep sea is the largest living space on our planet. It is a world of immense pressure and profound cold. More than anything, it is a world of darkness. Sunlight, the source of energy for most life on Earth, cannot penetrate these depths. Below a few hundred meters, the ocean becomes a realm of perpetual twilight. As you descend into the abyssal zone, the darkness is absolute. It is a blackness so complete our eyes would be utterly useless. For centuries, we could only imagine what creatures might live there. We pictured monsters and strange, formless beings in the eternal night. Yet life thrives here. A remarkable evolutionary story has unfolded in this dark expanse. Creatures adapted to the absence of sunlight and invented their own. This is bioluminescence. It is the production and emission of light by a living organism. In the deep sea, it is the rule, not the exception. An estimated 90% of mid-water animals have this ability. They turn darkness into a glittering cosmos of living light, a slow-motion, silent fireworks display. Light is not just for show, it is a fundamental tool for survival. Light is used to find food. Light is used to attract a mate. Light is used to defend against predators. It is a language. Flashes glow shimmering clouds. Understanding it reveals life in the deep ocean. These systems are alien, yet familiar. The challenges of seeing, hiding, and communicating are universal. Deep-sea solutions show evolution's ingenuity over millions of years. We will journey into that darkness to learn how deep-sea fish invented light. We will cover three main themes. First, adaptation. Why and how it evolved. Second, physical characteristics and anatomical structures. Finally, the hardware, the chemistry, and the physics. We will use scientific studies, archival footage, mathematics, and physics to illuminate this hidden world, revealing the elegant engineering of nature's own light creators. Imagine descending slowly into the ocean's midnight zone. The pressure outside your vessel builds with every meter. The water temperature drops to near freezing. The last vestiges of sunlight have long since vanished, and you are enveloped in a perfect, silent black. Then, a single point of light appears. It pulses gently, a soft blue beacon in the void. Soon, another joins it, then another. Before long, you are surrounded by a symphony of lights, strings of glowing dots, like runway lights on an invisible highway. Sudden, brilliant flashes designed to startle and confuse. Subtle, steady glows that mask a creature's silhouette from predators lurking below. This is not a fantasy. It is the everyday reality of the deep sea. The light you are witnessing is produced by the fish themselves. Each species has developed its own unique way of generating and using light, tailored to its specific needs for survival. For a small fish, a bright flash might be its only defense against being eaten, a photonic scream to stun a hunter for a precious second. For a larger predator, a dangling, luminous lure might be the perfect tool to entice a smaller meal into its waiting jaws. Light is the currency of this ecosystem, bartered for food, safety, and the chance to reproduce in the endless dark. How do you find food when you cannot see it? How do you avoid becoming food for something you cannot see? How do you find a partner in an environment that is effectively infinite and black? For the earliest inhabitants of this realm, these were matters of immediate survival. The absence of sunlight created a powerful selective pressure that demanded an innovative solution. It likely began with simple biochemical accidents, mutations that made cells emit a faint glow. In most environments, that glow is useless and costly. But in the deep sea, even a faint glimmer could give an advantage. A fish that glowed might better attract tiny prey. Bioluminescence evolved independently many times. It's not from a single common ancestor. Different groups arrived at similar luminous solutions. Convergent evolution. Simple models illustrate this. Imagine a population of deep-sea fish. A small percentage has a mutation for a very weak glow. That glow gives a 1% better chance of catching food and surviving. In the next generation, slightly more fish carry the trait. Another mutation makes the glow brighter or switchable. That gives a 2% advantage. Over thousands of generations, those tiny, incremental improvements accumulate. The result is the complex, controlled bioluminescence we see today, a product of relentless pressure to survive in the dark. One of the most common and ingenious uses of bioluminescence is for camouflage. This might seem counterintuitive. How can producing light help an animal hide? The answer lies in the world as seen from below. While the deep sea is dark, it is not uniformly black. A tiny amount of sunlight filters down, creating a faint deep blue glow from above. For a predator lurking below, any fish overhead appears as a dark silhouette. Visual. Top panel. Predator looking up sees a dark fish silhouette against the dim surface light. This makes them an easy target. To solve this problem, many deep-sea fish evolved counter-illumination. They use their own light to erase their shadow. Along their bellies are rows of photophores. Photophores point downward and emit a gentle blue light. The fish precisely control intensity and color to match the faint sunlight from above. Visual. Bottom panel. A predator looking up sees nothing, as a fish with counter-illumination blends into the background. The fish's silhouette disappears, invisible, cloaked in its own light. This isn't on-off, it's dynamic. Light from the surface changes with time of day, cloud cover, clarity of the water. Fish must constantly adjust photophore brightness. Scientists think photoreceptors on the top of their heads act as light meters. Those sensors measure ambient light from above, and the nervous system regulates photophore output. It's a living feedback loop, an automatic stealth system. Evidence is compelling. Studies of hatchetfish show placement and angular distribution of their lights perfectly cancel shadows across many viewing angles. Their light is spectrally matched. The wavelength matches filtered deep ocean blue. In submersibles with lights on, researchers see them clearly. With lights off, and only ambient light, the fish vanish. They are masters of disguise, using light to disappear. At the heart of bioluminescence is the photophore, the fundamental organ of light production. Most photophores share a basic structure, like a lamp or an LED. The core is a cluster of specialized cells, photocytes. These cells generate light via a chemical reaction. This cluster is the engine converting chemical to radiant energy. Around the photocytes is a layer of dark, pigmented cells. That layer is the lamp housing. It blocks stray light. The pigmented cup directs light outward, away from the fish's body. Many photophores have a reflective tissue layer made of guanine crystals. The reflector lines the inside of the pigmented cup behind the photocytes. It acts like a mirror or reflector dish. Backward emitted light hits it and is bounced forward. Finally, the structure is overlaid by a transparent cap or simple lens. The outer layer protects the internal parts. In complex photophores, the cap becomes a lens to focus or disperse light. Together, these parts form the blueprint for a biological light fixture, refined by evolution. The most remarkable of these are biological lenses. Just like the lens in a camera or telescope, these structures are transparent tissue shaped to bend light. A convex, outwardly curved lens makes parallel rays converge, focusing them into a tight, intense beam. That beam is perfect for a spotlight to find prey or signal far away. Hatchetfish photophores have U-shaped, angled reflectors. That geometry directs light downward in a specific pattern, brightest below the fish, fading to the sides, mimicking surface ambient light. Some photophores work like biological fiber optics. Scaleless dragonfish have light organs linked by long, transparent tubes. Those light guides pipe light from deep inside to points on the skin. These fish are masters of applied physics, using tissue to build advanced lighting systems. The color of light produced by deep-sea fish is a critical aspect of its function. The vast majority produce light in the blue-green part of the spectrum. This is no coincidence. Blue light travels the farthest through seawater. However, some fish evolved the ability to change color using specialized filters. They work by selectively absorbing certain wavelengths while letting others pass. The most dramatic example is the stoplight loosejaw dragonfish. They still have a main photophore that produces blue light, but in front of it, a special fluorescent protein absorbs blue and re-emits red. To use it, they evolved eyes with a chlorophyll-derived pigment that detects red. It's a completely private, secure channel of illumination. The loosejaw can cast red light like a sniper's scope to illuminate prey. Since most deep-sea animals can't see red, the prey stays unaware. It's the ultimate stealth hunting tool. At the very core of bioluminescence is a chemical reaction, a form of cold fire that generates light without producing significant heat. The process, in its most common form, involves two key types of molecules, a luciferin and a luciferase. The luciferase is the enzyme, the catalyst that initiates and controls the reaction. The luciferase molecule has a specifically shaped active site. It binds to a luciferin molecule, holding it in the perfect orientation to react with oxygen. The luciferin is oxidized, raised to a high-energy, unstable state. This excited state is fleeting. The molecule immediately wants to return to a more stable, lower-energy state, and it does so by releasing the excess energy. As a photon, a single particle of light. This process is remarkably efficient. While an incandescent light bulb converts only about 10% of its energy into light, bioluminescence can convert over 90% of the reaction's energy into light. Not all bioluminescent fish create their own light from scratch. Many have outsourced the job. These fish have evolved specialized sacs or organs that serve as living culture flasks for colonies of light-producing bacteria. The fish provides a safe, nutrient-rich environment for its bacterial tenants. In return, the bacteria do what they do best. They glow continuously. Since the bacteria glow continuously, the fish needs a way to turn the light off. Instead, it has evolved mechanical shutters that can be drawn over the light organ to block the glow. A young anglerfish is not born with its glow. It must acquire the correct strain of bacteria from the surrounding seawater. For fish that produce their own light, control is often exerted at the cellular level. When a fish needs a flash of light, its brain sends a nerve impulse down to the photophores. This binding event initiates a cascade of internal signals within the cell. This sudden change in the cell's internal chemistry is the final trigger, producing a burst of light. This neural control allows for incredibly rapid and complex light displays. This level of control elevates bioluminescence from a simple chemical reaction to a dynamic and expressive language. The invention of light was not just a chemical trick. It was the key that unlocked a new physical reality, allowing life to build a world of sight, communication, and complexity in the eternal night of the deep sea. First, we saw how adaptation was driven by the relentless pressure of the dark. Bioluminescence evolved as a powerful solution for finding food, hiding from predators, communicating with mates. Next, we examined the physical characteristics of these living lanterns. We saw how evolution assembled reflectors from crystals, shaped lenses from proteins, and arranged these organs into unique patterns that serve as a visual language. Finally, we delved into the hardware of light production, the chemistry, the physics, and the biology that make it all possible. This convergence of chemistry, physics, and biology is nature's ultimate engineering feat. Together, these three threads weave a complete story. Our exploration has illuminated much, but it has also revealed how much we still have to learn. The deep sea remains the largest and least explored environment on Earth. We are only beginning to decipher the complex language spoken in flashes and glows. What are the full vocabularies of these species? As we unravel these secrets, we find that nature's designs often mirror our own technological achievements. The photophores of a deep-sea fish share principles with our most advanced LEDs. By studying these biological systems, we can gain inspiration for new technologies. These creatures are not just curiosities, they are a library of proven engineering solutions. They remind us that nature is the original inventor. It shows us that even in the most crushing darkness, life finds a way not just to survive, but to create its own brilliance. As we continue to point our cameras and our curiosity into the deep, we are witnessing one of the greatest stories of invention ever told. A story that connects the fundamental laws of physics with the boundless potential of life itself. The dark is not empty, it is full of light.